This for Professor Herzenberg to look at in what direction.
What about you?
Well, I don't know. I defer on these things to this Friday morning, April 19th, 1991, we are in the National Museum of Health and Medicine of the Armed Forces Institute of Pathology on the campus of Walter Reed Hospital in Washington, D.C. We are continuing the story of the history of the cell sorter, which we started at the Becton Dickinson Immunocytometry Systems in San Jose, California, and then at the laboratories of Professor Herzenberg at Stanford University School of Medicine. We are standing in front of the AIDS education exhibit, which has incorporated the FACS2 cell sorter, which was developed at Stanford and Becton Dickinson and is now a part of this display. Professor Herzenberg will give us more detailed history of the early development of this machine later this morning.
Okay, let's do this while we're thinking of it.
That's what their newsletter is called at BDIS. It's just the facts.
The facts and nothing but the facts.
Okay, all right, everyone? Roll.
It's rolling and we're
Stand by.
We are now looking at the FACS cell sorter as it is displayed in the AIDS education exhibit. This was the instrument that was in the laboratory of Dr. Herzenberg at Stanford University. Dr. Herzenberg, how does it feel seeing your old instrument on display in a museum?
Well, a bit strange. I mean, it's still similar, similar enough to be recognizable as an instrument that's still being used, and even 20 years later. It's a bit musty here in a museum, and does look like it's dead, but it's a functioning machine.
How close was this model to the first commercial model that was built?
It's extremely similar. There's very little difference that's obvious. The electronics is different. There are some parts that were added after the machine was produced by Becton Dickinson, and so there are many added features to it in terms of detection, in terms of fluorescence wavelengths that can be dealt with.
We'll look at one of the panels that has a schematic diagram illustrating how the FACS works, and maybe you can tell us a little more detail the principles involved.
Okay.
Be quiet. We'll take.
It's rolling, we're ready.
We're looking at a very rough diagram, schematic diagram of how the FACS works. Dr. Herzenberg, could you give us a brief description of some of the basic principles involved?
Sure. We have a suspension of cells which have either come from blood cells or from some organ, tissue culture. So cells are suspended and put into this cell reservoir, it's called here, at point A. And then the cells will enter into the apparatus, into the nozzle, in the center of the nozzle, which is indicated as B here. Then there's a sheath fluid which surrounds the cells, and the cells are maintained in the center of the nozzle in the center of the stream that's coming out of the nozzle. It's forced out of the nozzle under some pressure. The next thing that happens is that the cells are illuminated by a laser, or it can be two lasers. Now, the cells will be detected by the laser if there is a fluorescent molecule on the cells. Either fluorescent molecules, either intrinsic fluorescence or more commonly antibody reacting with structures on the cell surface or with structures inside the cells. And these antibodies now are labeled with a fluorescent molecule. Fluorescent molecule means it's excited by the laser light and then gives off light of a different wavelength, a different color. And each type of cell is hopefully labeled with different fluorescent antibodies and giving off, therefore, a different color. With the modern machines, there are many different colors. With the original machine, it was just one color that we could work with. Okay, so now the cells come out of the nozzle in a stream. The laser is exciting fluorescence on cells in that stream. A little bit later on, the stream is breaking up into droplets because it's being shook by an ultrasonic vibrator up here. Causes the whole stream to be breaking up into droplets. And they're very uniform sized droplets, 30,000 or 40,000 droplets a second. So most of these droplets are empty, but about 1 in 10 droplets contain a cell. If the cell gave off fluorescence of the kind that we're interested in detecting, then as the stream is breaking off into droplets, a charge is placed on the stream. I don't see oh, there's a drop charging signal. It's placed on the stream. It's a drop charging signal. The drops are now charged. A few drops, 2 or 3 droplets, are charged, and then the charge is relaxed until another such droplet containing a desired cell is about to be formed. But now let's observe the droplets that are charged. They will continue to fall by gravity, and they will go between 2 charge deflecting plates at E here. The charged droplet there will be deflected by the positive or negative charge on the deflecting plates, and then will fall into a collecting tube, which is right here. And it can either go to the right or go to the left. Now, empty droplets and unwanted cells, droplets containing unwanted cells, will just fall into the collecting reservoir for unsorted cells. So to repeat, you can deflect one type of cell right and one type of cell left, because you're deflecting the droplets containing the cells, and the empty droplets or droplets that contain unwanted cells will fall in the middle. That's basically
The sort is completed.
Yes.
Thank you.
Now, the really important thing, so far as I was concerned years ago, our group was concerned, that we could sort live cells. Other people were interested in sorting dead cells, fixed cells, stained in various ways. But the essential for me was to sort live cells, because we wanted to sort these cells and then follow up properties of the cells. We wanted to grow them in culture. We wanted to see if they made antibodies. We wanted to see what other things they could do. And we were interested in sorting different types of cells. And for example, nowadays
But that's what I'd like to discuss here, so we'll stop here, because I want to...
Okay, tapes rolling.
We've looked at the schematic diagram, Professor Herzenberg. Now we're looking at the instrument itself. Could you describe some of its parts?
Surely. This instrument was at our laboratory at Stanford and has been shipped 3,000 miles now to Washington, D.C., where it's on display at the Smithsonian and Walter Reed Institute of Pathology. It's a bit shocking to see it here, but it looks like it's more or less the way it was when it left, except it's dustier. Okay, I talked about the cells being in suspension, single cell suspension, put into the tube. The cells now are forced up by some air pressure which comes from this area here, not very important. But the cells are forced up this tube, and behind this plate the tubes continue, and then they come out the other side, and there are some tubes which are not visible to you, and go into the nozzle. I'll remove this microscope, and I'll remove the protecting plate. The protecting plate is put there because there's a laser light, and it can be damaging to the eyes. And when you open this up, automatically the laser is shut off so that you won't be harmed. Now, the nozzle is right here. The stream is coming out of the nozzle, breaking up into droplets down below. The deflection plates are here. And then what's removed is the apparatus which has the collecting tubes and the reservoir for collecting the unwanted cells or empty droplets. So that you can imagine that the two tubes are here for the wanted droplets. Now, this area here is just things for controlling the laser beam. You can move the laser vertically, either roughly or coarsely or fine. You can move the laser horizontally as well as vertically. And it's mainly just a positioning of the laser beam so it aims right at the stream, which is a very narrow stream and would not be easily visible at the magnification that you're looking at it. So then the droplets are separated and the cells are collected below. This whole thing is a means of keeping the cells cool because we want to run sometimes for several hours at a time, and it can be run aseptically because this is usually in a clean room, and you don't get bacteria falling into the closed tubes which are down below.
That green gauge, what is that used for in the middle of the...
This? Well, that's just a means of positioning the point where the droplet is breaking off. You can raise or lower this apparatus, and this is a very fine micrometer for raising or lowering it. And you can see that if you look in the microscope, you have to look into here, and then put that over there, and now look in the microscope, and when it's working, you can see the droplets where they're breaking off from the stream, and you adjust it at a certain location.
And the other gauges are the pressure gauges.
So then over here we can see these pressure gauges, and there are pressure gauges for the sample, which controls the pressure on the tube, which contains the cells. And then there's a pressure for the sheath, it's called. The sheath fluid surrounds the place where the sample is being introduced, and by laminar flow, the stream, which contains the droplets, which contains the cells, is maintained in the middle of the stream, surrounded by the sheath fluid. And then these are just controls for the two types of pressure. And you're not supposed to touch this. It says on it, actually, I'll touch it. Can you read that? It says, don't touch. This is the way it was at Stanford, and you're not supposed to touch that because that's the main pressure valve, and it's always left on. Then there are a set of controls for stream deflection, left, center, right, deflection plates on and off, and there's a drive for the, all sorts of different little controls here. Now, I don't know if the camera can come over here, but if it can, this is the electronic control for the system, so that the signals would be collected by light detectors and brought into the electronic circuitry here, a lot of circuitry. And I'm not going to try to explain that, partly because even though we developed it, I'm not the engineer, and so I don't know all the details of the electronics, certainly. But this controls whether the droplets are deflected to the left or to the right, and how far they're deflected, and whether we're picking up signals for light scatter or for fluorescence. So that actually a very important part of the machine is embedded behind these panels. But the real guts of the machine is where we saw it, where the cells are introduced and actually deflected. And as I said earlier, and I'd like to repeat it, that a major objective for us with this machine was to be able to separate live cells. And that was not easy. We want to separate them live, and we want to keep them aseptic, so they are not contaminated with bacteria or other, say, molds. And we want to grow the cells and see what they can do in terms of making antibodies or other kinds of responses.
Now, was there a computer also that was attached here? What kind of computers did you use?
Well, originally there was not a computer. 20 years ago when we first built this, there was simply a kind of very primitive computer which collected the signals, and those signals were the data to go into this sort of computer. Then as the years have gone by, computer technology has improved enormously, and we now collect an enormous amount of data on every sample, store that data indefinitely. We first calibrate the machine and have beads for calibrating it. We compensate for overlap of different fluorescence signals. There's a tremendous amount of computer analysis and storage of the data, which is then put out. Now,
Well, thank you. We'll
Okay.
understand. Continuation.
okay.
Tapes rolling, we're ready.
So in the back of this instrument, we have a lot of plumbing, which is not normally dealt with, because in the front we had the control panel and the pressure indicators and so on, and back here is just the plumbing so that all the tubing gets connected, and finally you end up with the cells coming from the sample chamber into the nozzle and coming out and being sorted. We also see the laser, one of the early lasers, which generated, put out a signal, put out a light signal, which got bent around by prisms and mirrors in here, and came out to illuminate the stream. And we also see back here the photodetectors, and there are two of them at right angles. One was for scatter, light scatter, and the other one was for fluorescence in the original design of the machine. And then there are just cables here which go to the electronics part of the machine, which we've seen. And that's basically it.
Why this open design? Why not encase it in some...
Well, the objective was to have a machine which could be modified in many different ways, and it has been modified without a change in the fundamental design in 20 years. It's been modified enormously. So we've had many more photodetectors at different sides. We can change the, I can't get this out, but here it is. We can change the filters. We can change the mirrors. I don't know if they're in here now or not. It's a little bit stuck. But there are filters in here, and so we put more filters. We put additional lasers back here. We have 3 lasers planned for the newest machine. We have sort of 2.5 lasers now, so we have more capability of excitation and more capability because of the detectors of seeing the fluorescent wavelengths put out. And then it's open so that things can be maintained easily. That's it.
Thank you. You don't see the spots on the glass, or get the lights on the glasses? Is that doesn't show up there? It doesn't to the camera. Okay, all right.
The very top of the glass, not anywhere near his eye.
Look at Ray. Okay, who's that?
Yeah.
A number of times this morning, when you were describing the instrument, the fax, you made the point that what you were trying to do is develop an instrument that would look at live cells. Now, why was it so important for you to look at live cells? And was this an approach that was also being used by others who were trying to develop cell sorting instruments at that time? We're talking, what, mid-60s?
Mm-hmm. Yeah, well, our motivation was that we wanted to look at and then sort live cells so that we could do immunological experiments and genetic experiments. And that meant we had to have live cells sorted and then analyzed. This is much like biochemistry, where it was necessary to use gentle methods for separating different protein molecules or DNA molecules later on. We wanted to separate cells, which were the unit of body function, units of body function. Now, we had observed in the microscope that there were several different types of lymphocytes based upon their ability to bind different antigens, different substances which could be the instigator of an immune response. We really wanted to test the so-called clonal selection theory, and that meant that cells which bound a particular antigen should be able to make antibody to that antigen, and cells which bound a different antigen would make antibodies to that antigen, and that the further consequence of that theory, or further prediction, was that cells which bound antigen A would not in general make antibodies to antigen B, and vice versa. So that depended, in order to test that theory, we felt it was really necessary to sort type A and type B cells, and then have them live, have them be either put back into a mouse from which they came, or have them grow in culture and make antibody A or make antibody B, and then, of course, determine which kind of antibody was being made. That's why we wanted live cells.
Now, what were, and you went looking then for a kind of instrument to do that. Weren't there any other techniques available at that time to separate cells? Why weren't they useful for you?
Yeah. Well, we used whatever techniques we thought were possibly applicable, and we could sort cells based upon their electrophoretic mobility or upon their charge. We could sort cells on the basis of size, but we could not sort and recover cells on the basis of their antigen specificity. That is, there was at the time, there were a number of attempts to sort cells on the basis of antigen specificity without having to develop a cell sorter, such as we did. Such things as binding to solid materials, plastic supports or nylon supports, or even just petri dishes, where we tried to bind cells that bound antigen B or A by having antigen A or B on the plates. The problem was that we could bind them, but we couldn't get them off. Or at least we couldn't get them off in viable form, and nobody was able to get them off in viable form. And at the time, a number of different laboratories were trying to do just that and weren't succeeding. So I kept asking people who might have heard of a device or a means of sorting, separating cells which bound antigen A versus B or B versus A, And I did hear then that there was a machine at Los Alamos, which Mac Fulwiler had talked about, which could separate cells, and they could be live cells in principle. But it turned out that that machine only sorted or only separated cells on the basis of size, and size wasn't what I was looking for. And I also heard of a machine which was at the IBM laboratories in New York. And I was told that machine would separate cells based upon what it could bind. But then when I went to look at that machine in New York, I found that it didn't. And it only sorted very few cells, and that it didn't sort live cells, and also didn't even sort by fluorescence in general, although fluorescence could have been used. So these two machines were the two precursors I was told about and did investigate as possible precursors for the sorter. In fact, I learned something mainly from the Los Alamos experience, that you could sort cells and you could sort live cells, but that it was not done by fluorescence, and therefore it wasn't suitable for looking for specific antigen binding cells. But I went and saw both machines, and IBM actually gave me the machine with all its optical components and parts. Gave it to Stanford for me and my group. And we ended up cannibalizing that machine because there were many good optical components to it. But we really didn't make any use of the machine as such. It didn't serve our purpose, and didn't even sort cells at a reasonable rate. I mean, it only sorted a maximum of 300 cells, and that was it. And it was intended to be used for cancer cell detection, and that didn't require live cells. The machine at Los Alamos, New Mexico, which had been built by Mac Fulwiler, who deserves credit for having built the first sorter, even though he was not interested in fluorescence and didn't apply fluorescence to it prior to the time when we did that. It worked as a volume or size cell sorter. And Fulwiler, when I went to Los Alamos, I asked Fulwiler if he would consider putting fluorescence onto the machine, because I'm not an engineer and I just wanted to do the experiment, which was based upon, I thought, the only way of doing it was with fluorescence. And Fulwiler said, well, that isn't our particular interest, and we really cannot do this. So I asked him if we could have the plans for the machine, or I asked him actually first if he would build a copy of his machine, and we would try then to put fluorescence onto the machine. He said no, he couldn't do that. So then I asked, could he give us the plans of the machine, and I would try to get it built back at Stanford by some engineer or engineers. And he said, sure, we can give you the plans, and he gave us the plans. I gave them to an engineer at Stanford in the Instrumentation Research Laboratory, which was in our department at the time.
Was that Bonner, the engineer that you
No,
gave
it wasn't Bonner. It was an engineer who disappeared from the scene soon after he got the parts, so I don't think the story should involve him.
Okay.
But he got the parts, and he bought the parts, and he told me it was going to be $14,000 for doing it, and I got some money from Henry Kaplan and some from my grants with permission from the NIH to use it for this purpose.
Who was Henry Kaplan?
Henry Kaplan was a great cancer researcher and
At Stanford.
And he was a great man at Stanford. He was professor of radiology, the head of the radiology department, and had been interested in the thymus and in cancer, Hodgkin's disease. And when I talked to him about this kind of machine, he said, sure, he'd be very interested in it being built. And I knew he had sources of funds for the purpose, and so we did get that done. But then when that engineer just left Stanford soon after he bought the parts, and there I had the pile of parts on my desk, and not being an engineer, didn't know what to do, talked to other people who were in the instrumentation laboratory, and got Bill Bonner particularly interested in building a machine. And we used some of the parts and used other parts and adapted things. And for a couple of years, we worked together on trying to get this machine built. We finally did get a machine built, which would detect and then sort cells based upon fluorescence. That was the first fluorescence sorter. And we published that in Science in 1969, and you know the reference. Subsequently, then we made a much better machine, which still used the same principles, but was just electronically and engineering-wise considerable step above that machine, and that became the first FACS, the first fluorescence-activated cell sorter. That's the early history.
And you were at first using arc lamps, right, instead of lasers.
Yeah, Bonner's first machine was with the arc lamp. By the time we developed what we called the first FACS, that had a laser on it. And it's much like the current machine, except it's just bigger and just more capabilities, but it still is fundamentally the same machine.
Now, to flesh out the context a little more, you mentioned the instrumentation laboratory. That was set up for a very specific purpose and programming. Maybe you could talk a little bit about that.
Well, it was set up with funds from NASA, the space agency, with the inspiration and the stimulation and support of the chairman of the genetics department. It was Joshua Letterberg, who was interested in finding whether there was life in outer space and to examine the possibilities of that. And he wanted to build various kinds of instruments for that purpose. But he also had the idea that building an instrumentation research laboratory would have many functions for science here on Earth, in addition to the exobiological. That means exobiology was the study looking for life in outer space. And he was very happy that we were making use of that facility, and people in it were loaned to us. And NASA was given reports every 6 months on the progress, on our progress, as well as the progress of other projects. And so the early work was supported by NASA. I think that was working from 66 or so until about 69. And then we got an NIH grant because we had already published or had a paper in press showing that we could sort cells without killing them. We could sort viable cells. We could sort viable antibody-producing cells. And so that demonstrated capability. And there was a program at the National Institutes of Health for automating the clinical laboratory research program. We applied for money under that program, and we got it. And we then had our own support and continued working on it. And there's a long story involved, but I think the most important parts of it are that we acquired the services and hired him in our group, actually, the services of Richard Sweet, Dick Sweet, who just so happened to have invented the separation principle of the droplet separation principle used in the sorter and used by Fulwiler, although he didn't know Sweet, and had been patented and developed as an inkjet printer. But it also sorted droplets, and the droplets could contain viable cells within them. And I've explained how the sorting works. There's still that same principle which is used to this day. And Dick Sweet still works for us, improving these machines. We've gotten a much larger staff, other engineers, other grant support. A lot of computers are into the project now. And we have a facility where at Stanford more than 100 different investigators make use of these machines for sorting cells, for analyzing cells, cell populations. And of course it's used very much these days for the study of AIDS, trying to investigate the causes and trying to investigate or determine the prognosis and how badly somebody has the disease. It's on the progress of the disease. And so in clinical laboratories throughout the world, these cell sorters have their application in studies of blood cells, and particularly a very important application is for AIDS. And I believe that's why it's in the middle of an AIDS exhibit here at the Armed Forces Institute of Pathology, and why the Smithsonian, to whom we gave the machine, the Smithsonian Institution, has loaned it to this particular exhibit, because as I said, it does have a tremendous relevance to the study of AIDS.
Going back to going back a little bit to the early history, like after 69, early 70s, once you've had a prototype model built and working in your laboratory, how did you go about trying to get a commercial company interested in producing a model, a working model for the laboratories?
You know, as I'd said, we had an NIH grant, National Institutes of Health grant, to build a machine and to develop applications for the machine. And of course I was publishing and giving talks about work with the machine. And there were undercurrents and then overcurrents of concern, criticism that we could do these experiments, but there was the only machine in the world which could do this kind of experiment, various kinds of experiments. And my colleagues, other workers in the fields of immunology or genetics, wanted to have such a machine, and so I started looking for companies with the help of officials at Stanford, try to find a company that would be willing to produce the machine. I did have one bite which seemed to be working, and Dick Sweet was the person who worked for that company, who had the assignment of trying to develop a machine with us. But after a few months, that company took itself out of the picture, no longer was interested, and that was Varian. And the main contribution they made was that Dick Sweet was willing, more than willing, to move from Varian to Stanford. Since I had grant support, I could hire Dick Sweet as an engineer to further work on this project, which actually was very interesting to him. So he came over to Stanford, and out of that we developed a prototype, and a good prototype. Then came President Nixon's war on cancer, so-called, in which he declared that cancer will be cured in 10 years by presidential fiat, presidential decree. And a lot of money was made available for this project, and scientists were brought to Washington and gathered committees. And long and short of that story was that the immunology program at the NIH was told they had X amounts of money, I think $10 million, I believe. And the head of the immunology program at the NIH went around the country talking to various immunologists, and I was one of them, asking how this money might be spent effectively in the cancer war. And I said, well, you know, I can't imagine anything more useful at the moment than to build a number of these machines, or to start the ball rolling to building a number of cell sorters. We had one, but that was it. We didn't have the means yet to produce any more. And I said, why don't you form a committee and have this committee consist of scientists who are knowledgeable about immunology and also knowledge about various areas of science and engineering. And different people can apply for grants for much larger amounts of money or contracts for much larger amounts of money that are normally available under the grants program. And he thought that was a good idea. And I said, we'll apply for one of those contracts, and it will be to build 2 machines, one which will be in our lab and one which will be in your lab. And he said, that's a great idea. And so that was done, and we got a contract agreed to by a peer group. And we then looked around for a company which would be willing to take the contract and build the machine. And I guess Varian was approached again, but they were not interested, as I didn't think they would be. And it happened to be that I was an advisor, consultant to Becton Dickinson at that time, as an advisor in immunology. And I told them about the machine and told them that I thought it had tremendous potential. They didn't think it had much commercial potential. In fact, I think they thought that maybe if we built 10 machines over a long period of time, that would be the market. I thought that was wrong. That they could build 30 and be able to sell them. But it was still, we greatly underestimated the potential market for these kinds of machines. In any event, we had the contract, and we subcontracted it to BD, Becton Dickinson, and an engineer was hired to undertake this project under the direction management of Bernie Shore, and with Dick Sweet of our lab playing the coordinating role in seeing that they built a machine which could do what our machine could do, but it would be much more compact. And it is, in fact, very much like the machine which we just saw. That's, I think, the backdrop to this conversation. So that machine was built, 2 of them were built, and then subsequently they would build more machines for colleagues of mine who wanted a machine with the same capabilities. And they built them one by one until the business built up, and eventually they developed into a company of some several hundred or 700, I think, people, based upon the fax machine and based upon a later development of monoclonal antibodies, which was very important for exploiting the potential use of fax machines. That's the story.
What was it like, this joint venture with a commercial company? We hear much about the differences in the way science is done in academia and in the corporate world. How do you see that today?
Well, there was a small unit of this major corporation, Becton Dickinson. The head of this small unit of engineers really was just an engineering, well, I shouldn't say just, it was an engineering group. And Bernie Shore took on the project, hired this engineer, David Capillaro, and they worked closely with us to get the machine, but it was at no cost to them. It was a contract paying for the total cost of building the 2 machines. They made their whatever profit they made was built into the contract. They got paid overhead. So it really was a cost-free, no investment required to get started. And after they got started, then they would take orders, and they would get in a deposit for building another machine and building another machine. And then I went around with Bernie Shore to different immunologists and geneticists, and basically served to publicize the existence of the machine, the possibility of getting one built. And Bernie Shore was sure to get the money to build the next one from the last one and from a deposit. So it was a no-risk venture and worked very successfully for a while. And then they realized that they had a product which had a greater potential, and particularly when they learned about monoclonal antibodies, that that this machine was required for being able to sell large amounts of monoclonal antibodies, which fit in with their business a lot better. They expanded, and then they got someone else who was capable of directing the expansion when Bernie Shure was thinking of retiring, or I don't know whether he's retired yet, but in any event, they invested some money in that and are doing very, very well, I think. They have now something like 700 employees. They have an enormous building, and it's a successful venture. But I found it very difficult, to be very frank, to see that we had different objectives than the company. The company was interested in a mass market of some sort, and we were interested in quality machines which could do more and better, more useful things. And there has been over the years a dichotomy of our interests at Stanford and the interests of the company. But we've managed to, in general, work out a means of operating which would build good machines and reliable machines, not as good and not as capable as we knew could be built, and we continue to build more capable machines at Stanford. And for a while, the company followed our lead, but then they found that they didn't want to follow our lead anymore, and they wanted to go into a market where they could have simpler machines which weren't as capable but could be used in clinical laboratories much more easily, so less openness of the machine. It would be enclosed in a box, and the guts of the machine would not be accessible to technicians or technologists or doctors who would be using the machine. And that was the opposite way that we wanted to go, where we wanted to have the most capabilities that were possible, and we wanted to have the machine adapted to all sorts of different purposes by researchers. And maybe now there is going to be some kind of a rejoining of forces, but I don't know.
Does Stanford hold the patents on most of these machines?
Stanford has a patent on the machine, which is about to run out. Patents only last for a certain number of years, and then they're free. And BD has paid Stanford a royalty on sales of fax instruments, and that royalty has gone to Stanford University, and it's been divided between the Department of Genetics, the School of Medicine, and our laboratory. And we've made good use, I think, of the monies that have come in under those patents, under the patent, and under the royalty agreement, so that we've actually been able to, well, for example, buy the computers that have allowed us to go into computer developments for the machine. And we've used it basically to support the research activities under our general direction in both on cell sorters and on immunology and genetics and developing new applications. And really the work over the last 20 years in my laboratory has been partly supported by that. Of course, it's been majorly supported. I mean, the major support for the lab has come from public funds, from the National Institutes of Health in general. And
as you know, many major scientists in the biomedical field have now gone out on their own and formed companies, and at the same time keeping some academic affiliations. How do you feel about that, and were you ever, or are you still tempted to do that yourself?
Well, the answer is no, that we're not tempted to do that. I mean, I think I've learned enough in my lifetime to know that I like working with the freedom that the university provides. I know that we have to scramble for grant support at times, and sometimes that's uncomfortable, particularly when funds are short. But we've managed to survive, and more than survive. And I like having students. I like having postdoctoral fellows. I like being able to work in whatever area we intellectually choose to work in, if we can get the funds to work in. And I like having also the extra funds that, the extra flexibility and use of funds that were given to us by this patent agreement. It's only a fraction of the money that Stanford University got, but it's a very useful fraction. It's allowed us to take on postdoctoral fellows when we didn't yet have support for these fellows, and sometimes to pay them for 2 or 3 years. It's allowed us to increase our engineering staff and be sure that we had money to pay them. The other thing is that I've made a certain amount of private money, which hasn't changed my lifestyle, hasn't changed what I want to do with my life as I continue, but I've not been tempted to go out and actually work for a commercial company to become a president. I've been offered the presidency of Becton Dickinson Immune Systems when it first started, and several times afterwards, and never really got tempted because I prefer the academic freedom to work on what I think is interesting to work on. And right now we're working on AIDS ourselves, and it's beyond the scope of this interview, I think, to talk about specifics there, but again, I'm looking for federal support, public support to do this work, and I will use some of the accumulated reserves which we have from the patent income. And I think we're very lucky that we had this patent income, but it's going to end now. Becton Dickinson's going to cut off any further support for our laboratory through this patent mechanism. They might generate some more support, or we might generate more support through a development of computer workstations directed at fax kinds of studies as their first project. In fact, my wife, Lee Herzenberg, is trying very hard to get these workstations established, and it may turn out usefully for the next generation of instruments and of work that we do in our laboratory.
Well, maybe now we can take a broader look at AIDS research and AIDS education by taking a look at this exhibit and how the fax machine fits into it.
I think that's a good idea.
Everyone stay put. What I want to do is for everyone to be quiet. Let's just roll for about 30 seconds.
Okay, the AIDS exhibit within which the fax machine is contained now at this Institute of Pathology illustrates very clearly what the fax does for the AIDS problem. I mean, it counts the number of these helper lymphocytes, T4 or CD4 helper lymphocytes, which are present in the blood. Counts the number per cubic millimeter. Normally, one has from 500 to 1,000 of these cells per cubic millimeter, and as one progresses towards AIDS, they get fewer and fewer until towards the last stages of AIDS, there can be almost none or just maybe 10 instead of the 500 to 1,000 T4 or CD4 lymphocytes. So the virus is present, it's illustrated, and the virus will bind to a specific receptor, which is not illustrated here, on the surface of these T4 helper lymphocytes. Now, the second thing that the fax machine can do is to determine the amount of receptor per cell. So there might be 100 or 1,000 or 10,000 receptor molecules on the surface of each helper lymphocyte. So you can measure or count the number of helper cells, T4 helper cells, and you can count the amount of the receptor on the surface of these cells. You notice that it is a receptor because as we progress over here, that the virus is bound to the surface of the cell. It opens up, releases the RNA, which is inside, and then the life cycle proceeds. And in this last diagram, in this particular part of the exhibit, it points out that the AIDS virus, or the RNA from the virus, which is now introduced into the DNA of the cell as DNA, so it's a retrovirus. A retrovirus means that it starts out as an RNA virus. The RNA then is converted to DNA and is incorporated into the DNA of the cell. Now, these T4 cells and other cells, monocytes, I believe, have the retrovirus incorporated into them. And we can tell how many of those cells are present by use of the fax machine. So the prognosis, the expected fate of people with the disease, and what's more, the treatment of the disease, and there are treatments now which can increase the number of helper lymphocytes, of T4 helper lymphocytes, and offering the prospect that one day it might be possible to have a normal lifespan, even with the virus still present in these helper lymphocytes. I think that's cut.
30 seconds. I've got the camera mic on too. I have the camera mic on.
50th anniversary of
Tom, did you get the?
Yeah. Okay, so you just want the name, you don't want to see the...
It would be nice if it was flashing, of course, because if it was flashing, it would show it in the other thing, right? So why don't you pick it up here? Hold it for license, make sure it's there. If you did about anything else, we'll do it at the next campus. Okay, you got that?
I see she's part...
Okay, now it's the last one. How are you handling that?
Well, you gotta do a tilt down. And then like this, you gotta... Actually, they'd be interested in seeing condoms in 50 years.
Have you get a decent touch on it? I'd be kind of interested in that.
Chic Elite. There's nothing sweeter than safe sex. going to probably look at this in 50 years and say, radioactive material, that's what happened to us all. Those schools thought we had tape on. I was just focusing. Just stick your hand in there and hit one of them.
Oh.